Welcome to our educational video on cell injury. Normally, the cell is in a state of homeostasis. When an injurious stimulus occurs like ischemia, infections, or toxins, this balance is disrupted, leading to cell damage. It can be reversible when the stimulus or stress is removed. If the damage continues or becomes severe, we move to irreversible injury, which means that the damage has gone beyond point of no return. The cell can no longer recover even if the stimulus is removed. From here, the process leads to cell death. This is where the cell's fate is sealed, and it can proceed via two major pathways: necrosis and apoptosis. Reversible cell injury is characterized by changes that can be reversed if the injurious stimulus is removed. The morphological changes seen are cellular swelling and fatty change. In cellular swelling, the cells can be seen as vacuoles under microscope due to intake of water, leading to cytoplasmic vacuolation. It causes organ pallor, increased turgor, and weight due to widespread cellular swelling, and small clear vacuoles are seen under microscope, which are segments of distended endoplasmic reticulum. The next morphological change is fatty change, in which there is appearance of lipid vacuoles filled with triglycerides in the cytoplasm. There are also some additional intracellular changes in cytoplasm: plasma membrane, mitochondria, endoplasmic reticulum, nucleus, and appearance of myelin figures. Until this point, the changes can be reversed. But if the damage is severe and progressive, it leads to irreversible cell injury. It is the stage where injury progresses beyond the point of no return, leading to cell death. The cell death is by two ways: necrosis and apoptosis, which are discussed separately in other videos. Next, let's discuss the causes of cell injury. Cell injury can result from various stimuli, which are broadly categorized into hypoxia and ischemia due to reduced blood supply, toxins that can damage cells either directly or through metabolic pathways, infectious agents like bacteria, viruses, fungi, and parasites causing cell injury through various mechanisms. Immunologic reactions can lead to cell injury either through direct attack, inflammation, genetic abnormalities leading to cellular dysfunction, and nutritional imbalances and physical agents can also lead to cell injury. The mechanism of cell injury. The cellular response to injury depends on the type, duration, and severity of the stimulus, as well as the cell type, status, adaptability, and genetic makeup. Cell injury typically results from functional and biochemical abnormalities in essential cellular components, and we will learn them one by one. Hypoxia and ischemia result from insufficient oxygen supply, leading to cellular energy failure primarily due to the disruption of oxidative phosphorylation in mitochondria. Without oxygen, cells can't produce ATP efficiently via oxidative phosphorylation, leading to an energy crisis. Cells then rely on anaerobic glycolysis, which generates less ATP and results in lactic acid accumulation and decreased glycogen, lowering intracellular pH and impairing enzyme function. Cells activate compensatory mechanisms for survival through hypoxia-inducible factor 1, promoting angiogenesis and switching metabolism to glycolysis for energy production. This adaptation helps cells survive low oxygen conditions but is less efficient. Prolonged hypoxia leads to cellular damage, failure of ATP-dependent ion pumps, leading to sodium influx and potassium efflux, causing cell swelling, loss of microvilli, and blebs appear. Loss of ATP due to hypoxia and ischemia leads to cell swelling, detachment of ribosomes, and disruption of protein synthesis. Hypoxia might increase reactive oxygen species, especially during reperfusion, causing additional damage. Heart muscle stops contracting quickly upon ischemia, illustrating the severity based on the duration and extent of oxygen deprivation. Ultimately, severe or persistent hypoxia leads to necrosis or apoptosis due to irreversible damage to cellular membranes. Ischemia or perfusion injury occurs when blood flow is restored to tissues that have been deprived of oxygen, leading to further damage rather than recovery. When oxygen returns, it increases the production of reactive oxygen species by damaged mitochondria, which can't fully reduce oxygen. This exacerbates injury because the body's antioxidant defenses might be weakened by the prior ischemia. Reperfusion can worsen inflammation by bringing in leukocytes and plasma proteins. These leukocytes can release substances that damage cells further. The body's complement system, which is part of the immune response, might get activated, binding to injured tissues or antibodies there, leading to additional tissue damage through inflammatory byproducts. This injury is particularly significant in organs like the heart, causing myocardial ischemia, and brain, causing cerebral ischemia, where it can lead to cell death and further complicate recovery. Oxidative stress refers to cellular abnormalities caused by reactive oxygen species, ROS, which are free radicals. It is caused by unstable free radicals, damages cells through various mechanisms including necrosis, apoptosis, and necroptosis. They arise from chemical and radiation injury, hypoxia, cellular aging, inflammatory cells, tissue injury, and ischemia or perfusion injury. Free radicals with unpaired electrons react cellular components, nucleic acids, proteins, and lipids, propagating damage and leading to cell injury. During normal cellular metabolism, particularly in mitochondria, oxygen is partially reduced, leading to the formation of reactive oxygen species like superoxide and hydrogen peroxide. These are byproducts of the electron transport chain, where oxygen is eventually reduced to water. The superoxide anion is generated during mitochondrial electron transport. It can also be produced enzymatically by xanthine oxidase and cytochrome P450. The enzyme superoxide dismutase converts superoxide to hydrogen peroxide, and the hydrogen peroxide is reduced to water enzymatically by catalase and glutathione peroxidase. Hydroxyl radical is formed by two ways in biologic processes: by radiolysis of water and by reaction of hydrogen peroxide with ferrous ions, which is called Fenton reaction. In the immune response, neutrophils and macrophages generate reactive oxygen species through the respiratory burst to kill pathogens. Here, superoxide is produced and converted to hydrogen peroxide, which then interacts with myeloperoxidase to form hypochlorous acid, a potent oxidant. Nitric oxide produced by various cells, which can combine with superoxide to create peroxynitrite and other reactive species involved in cell damage. These free radicals cause lipid peroxidation, protein cross-linking, and DNA damage, leading to cell injury, cell death, aging, and malignant transformation. Next, cell injury caused by toxins. Toxins, including environmental chemicals and substances produced by infectious pathogens, induce cell injury that culminates primarily in necrotic cell death. Different types of toxins induce cell injury by two general mechanisms. Direct-acting toxins directly bind to critical cellular components, disrupting essential functions. These include mercuric chloride, anti-neoplastic, chemotherapeutic agents, and microbial toxins. These toxins inhibit ATP-dependent transport, increase membrane permeability, and target host cell molecules for essential functions. Many toxic chemicals are not intrinsically active but must first be converted to reactive metabolites, which then act on target cells. Latent toxins are activated by cytochrome P450 in the smooth of the liver and other organs. Carbon tetrachloride, for example, is converted to a toxic free radical principally in the liver, and this free radical is the cause of cell injury mainly by membrane phospholipid peroxidation, particularly damaging to hepatocytes. Free radicals released by carbon tetrachloride results in impaired enzyme and protein synthesis due to endoplasmic reticulum damage, dissociation of ribosomes from rough endoplasmic reticulum for impacting protein synthesis, accumulation of lipids in cells due to impaired apoprotein synthesis leading to fatty liver, mitochondrial damage, ATP depletion, and subsequent ion imbalance causing cell swelling and death. Acetaminophen, when overdosed, it similarly produces a metabolite that can severely harm the liver through similar mechanisms of oxidative stress and cell injury. Accumulation of misfolded proteins in the endoplasmic reticulum can lead to cell death by apoptosis. If unfolded or misfolded proteins accumulate in the endoplasmic reticulum, they first induce protective cellular response that is called the unfolded protein response. Unfolded protein response is protective cellular response that activates signaling pathways to increase chaperone production and decrease protein translation, thus reducing levels of misfolded proteins in the cell. When misfolded proteins accumulate and cannot be handled by the unfolded protein response, propaptotic signals activate BH3-only family sensors and caspases, leading to apoptosis. Misfolded protein accumulation causes gene mutations, aging, infections, increased demand for secretory proteins, and changes in intracellular pH and redox state. Protein misfolding can lead to disease by causing protein deficiency or inducing apoptosis. This process is linked to various conditions including cystic fibrosis, Alzheimer's, Huntington's, Parkinson's, and type 2 diabetes. If you found this video helpful, please like, share, and subscribe.